All right. Do you guys know what the National Academy of Sciences is? No? Yeah. Anybody? No. Oh, thought you were raising your hand. You're got a scratch, got an itch. Um, the National Academy of Science is a group of scientists who are nominated by other members of the National Academy of Sciences, and they are among the most accomplished scientists in the country. That's how you get nominated and elected to be a member of this group. Um, they come from all fields. There are a limited number of ecologists who are National Academy members, physicists, chemists, biomedical people, um, some molecular biology people. All of the sciences are represented. And what they basically do is, other than being really, really smart, is they will occasionally put together studies. And so if you go onto our website, oh, that's marks on the screen from last time I was here. Can't believe people just right on the screen like that. Um, we are going to start talking today about genetic engineering. So we've covered basic genetics, and I'll ask you some questions about the screencast here in a few minutes. But we're finished kind of with basic genetics, and we need to talk about genetic engineering because genetic engineering, for whatever reason, just scares the crap out of people. So in order for you to evaluate whether or not those fears are founded, you need to know something about genetic engineering, what it is, how it works, etc. And then you're going to read, and I want you to get started on this early, you're going to read portions of the National Academy of Sciences report. Because your assignment leading up to Thanksgiving, so we're starting this well in advance of when this is due, because I want you to have time to think about this. There is a position paper that you need to write, and this is described in the syllabus. If you haven't been back to look at the syllabus in a while, you should perhaps go and revisit that. And it's a position paper that is written in the form of a letter to your relatives. I don't know about you, but over the last couple of years, my relationships with my relatives have become more strained over just weird stuff that they say and think. And so imagine, the letter is basically written to imagine that you have a relative who is at Thanksgiving dinner with you, and this relative says something that indicates to you that he or she is fearful of genetically engineered crops being in their food supply. And the purpose of the letter is for you to inform them what the science is about genetically engineered crops in our food supply, and whether or not you think that there should be anything to be worried about in all of that. And the basis for fact for the purposes of this letter, explaining kind of what these things are, what effects they have, etc., is going to be this National Academy of Science report. So I think did I just download it or did I not just download it? I think I didn't. I'm not going to ask you to read the entire report. The entire report is overkill. But what I am going to ask you to do is I want you to read 4 chapters of it. So once you get all past all the preamble, some of which you should pay attention to. You should pay attention to who wrote the report. So it lists all of the authors, all the contributors to the report. It tells you what their affiliations are. You will find that a lot of the affiliations are academic researchers, but you also have representatives from industry. So you have representatives from Monsanto, Archer, Daniels, Midland, those types of agricultural companies. And so you're going to read 4 chapters. You're going to read chapter 3, which is kind of a history of genetically engineered crops from when they started genetically engineering crops up to the year 2015, which is when this report was written. So the report is a little old, but it's not terribly old, but it's still pretty relevant. Then each of you need to read chapter 4, chapter 5, and chapter 6, because it basically talks about 3 big categories of potential effects of genetically engineered crops. Agronomic and environmental effects, the effect on agriculture and the environment. Human health effects, which is I think what scares people most. And then something that most people probably don't think about is social and economic effects of genetically engineered crops. So this looks at genetically engineered crops from the perspective of the farmer, rather than from the perspective of human health or how agriculture, crop yields and stuff like that is concerned. You can read any of these other chapters if you want to, but that's the bulk of what I think you need to know to speak intelligently about this. So everybody needs to read chapters 3, 4, 5, and 6. So get started on it now. What I'm going to do probably by the end of this week is I'm going to assign you randomly to a set of groups. Within each of these groups, some of you are going to be assigned chapter 4, some of you are going to be assigned chapter 5, and some of you are going to be assigned chapter 6. Your task within your groups is to read those chapters really thoroughly and prepare a 15 to 20 minute presentation to your classmates of the information that is in that chapter. So you still need to read the other chapters, but I found it productive in the past. I do this in my biology classes. It's productive to schedule short presentations where people present to their peers information to kind of boil down complex information to the basics, the salient points that are made in each of these chapters. Your classmates can then take notes on that, and then when they go back and reread those chapters, their reading is a little more focused than it would have been had you not done this for them. So over the period of a couple of days, we'll have presentations of you guys presenting as a group to one another, to kind of get information across. That doesn't mean that, oh, I'm not in the group that is reading chapter 4, that's presenting on chapter 4, so I don't have to read chapter 5. I'm not in the group that's presenting on chapter 4, so I don't need to read chapter 4. You still need to read chapter 4 because your letter to your relatives has to have chapter 4 material in it. It's just that the chapter 4 people are hopefully working with me in doing a good job of presenting that information to you. So it's up on the Moodle site. Download it. Begin the process of reading it. The stuff that we're going to go in today will start to become relevant to you as you start reading, especially chapter 3 of this chapter, of this book. So presumably these National Academy of Science people don't really have any agenda except getting the correct information to policymakers and the public. For a book that is designed to be oriented towards policymakers and the general public, it's fairly technical. I'm not gonna lie to you, which is why you should get started on it now rather than putting it off, which is why I'm giving it to you so much in advance of when you actually need to start writing about it. One of the goals of the next couple of days is to give you enough information about genetic engineering that the technical things that you're going to be reading in the report are less challenging to you. Questions about that? Once we get past the presentations, then I'll set a due date for the paper, and so that will be coming down the pike. And fairly shortly here, I'll be setting a due date for when you need to have your presentations ready. Now, how many of you have made presentations while at Jewel? Okay. Have you ever been taught how to make a presentation? Yeah, you have? What'd you learn?
Speech, I did a speech class, so they don't have any confidence test, but just getting in front of a class and learning how to do that.
Okay, yeah, there are things that you should and shouldn't do, and we'll go through those as we get closer to presentation time. One of the things that you shouldn't do is you shouldn't have a lot of text on a slide in really small fonts. You should do as much as you can with images and things like this. If you've been watching me present information to you, do you see a ton of text on the slides? No, not typically. Um, some students don't like that. They're like, I want all the information typed out on the slide. I'm like, well, that would be terribly boring because then I might as well just give you slides with a bunch of text typed out on it and then just like go away, come back at the end of the semester and give you an exam. That wouldn't be terribly useful. So the more you can do with images and figures, there are figures throughout the chapters that illustrate the points that the people who are writing the chapter were trying to get across. Those are good figures to include in a presentation because it makes the point that the scientists are trying to make. It doesn't mean that you can't have any text. It just means that you shouldn't just have slide after slide of nothing but text. Yes?
Another point I remember now in speech class was don't just read from the slide.
Oh, yeah, don't just read from the slide. You might as well just shoot us all in the head.
Yeah.
Yeah, because we'll fall asleep and nobody will get anything out of that. So anyway, your assignment ongoing is start working your way through the National Academy of Sciences report, beginning with Chapter 3 and proceeding to the end of Chapter 6. For me, I have already thought a lot about the information in Chapters 1 and 2. So the first time that I read this report with my students shortly after it came out, I was most surprised and pleasantly informed by the information in Chapter 6 because I'd actually never thought about, I thought about the economic plight of farmers in the past, but I've never thought about that in the context of what genetically engineered crops have meant for the economic plight of farmers. So read it with an open mind, and if you're like me, you will learn something as you go through the thing. I actually learned quite a lot going through it, and I already knew quite a lot going into it. So that's coming down the pipe. Just be aware of it. You notice that I don't have any names on the Moodle site here because I haven't assigned you to groups yet. But as soon as I get that done, it'll appear on the Moodle site. All right. Let's go. Any questions from the screencast? About the what? Oh yeah, 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 sure. So let me actually go back to this. So imagine that instead of this being height, imagine that this is oil content from low oil content to high oil content. Low oil content to high oil content, once again, is corn oil in corn. So imagine that you're a farmer and one of your goals is to create corn oil. So we grow corn for all kinds of reasons. A lot of times we're growing corn for the carbohydrate content of corn, but different parts of the corn have different substances in it. So the little developing embryonic corn plant that is growing in the corn seed when you plant it, there's a thing called the embryo. We haven't gone through seed structure yet, but we will. That's where the oil is found. And so the oil is found in the developing new corn plant. Carbohydrate is this sugar and starch is what makes up the bulk of the corn. And then all of that is wrapped up in a seed coat. And the seed coat that goes around the corn is where you find any protein that is in the corn. So in a corn seed, the corn has different substances that are useful for for food scientists, has different substances depending on where exactly you are in the corn kernel. And when corn goes off to a processing plant, they basically break up the corn kernel. The seed coat goes one place, the starch goes another place, and the embryo goes a third place. And when you get enough of these embryos, billions and billions of these embryos, because they're very tiny, built up into a stock, you can basically crush those. And when you crush those, you can squeeze the oil out of them. And so the oil content that is in a corn kernel is a heritable trait. It's a trait that has variation from individual to individual, and it's heritable because it's also underlain by genes that affect oil content. So if you imagine that you have oil content from a little bit of oil per kernel to a lot of oil per kernel, you, the farmer, could actually select for higher content oil by just only allowing those plants that have high oil content to reproduce. So you artificially select against low oil content plants. And what that means is that all these red plants won't reproduce, all of these black plants will reproduce. That changes the average oil content of the plants that you're allowing to reproduce so that their offspring also have a higher oil content. This is just the reason that this thing is called the breeder's equation that's on the next slide is because it's an equation that was developed to help animal breeders improve their crops, improve their cattle production or milk production or whatever it was that they were trying to maximize. So this shift is actually selection, and this shift here is the change in the phenotype, or the evolutionary response to selection. So now imagine that you're doing this. Oh, and the degree to which this changes is dependent on the slope of this line. So notice how it increased a certain amount here, but it increased less here because the slope of the line is less than 1. And so this is heritability is the slope of the line. If it's the slope of the line is 1, then whatever you change in parents, that will be exactly felt in the offspring population. But heritability is never 1. It's always something less than that, and it can vary quite widely from down close to 0 to up close to 1. This is actually an exceptionally high regression for height in humans. It's usually down around 0.8 or so. It's just in this particular sample of students 1 year in my class, this is what it happened to be. So now imagine that you do this. Oh, so this is the breeder's equation. This breeder's equation just comes from how much selection are you exerting against the population? What's the heritability? That tells you what the response is. So it comes directly from how much selection are you exerting on the heritability is there. That leads to the evolutionary response to selection. And delta Z is the change in the trait, but it's also, oftentimes people just call it R, which stands for the response to selection. So if you exert selection such that you prevent one part of the population from breeding, what will the result or the response to that selection be in the next generation? And you can actually just do the math. If you know what the selection is, you know what the heritability is, you can tell how much the population will respond. So that's how that works. But that's just for a single generation. So imagine that you do this for 100 generations instead. So in this plot, at this farm at the University of Illinois, they basically started with a common population of corn plants. One set of plants, they artificially selected only the highest oil producing plants. And in another group of the crop, they selected only the lowest oil producing plants. When they did this, in the continuous high oil selection, they were selecting for high oil content. Oil content went up and went up and up and up and up, and it just kept on going up. In the low oil content, it went down, but you notice that it's going down at a slower rate than this is going up. Not only that, that rate is decreasing. The way you figure out what a rate is for a curve is you plot the tangent to the curve. And as you can see, as time goes on, the tangent to this curve is getting flatter and flatter and flatter. So the rate is decreasing, and it's decreasing at a decreasing rate. Whereas this is relatively constant. The tangent to that curve, it isn't even really a curve, it's just a linear line going upward. And so the slope of these lines that are tangents to this curve is getting less and less severe over time. And the reason for that is that there's just less variation. Because you're starting out at only 5% oil content here at the beginning, there's probably not a lot of dominant alleles. So remember that these are the kinds of traits that are determined by lots of different genes at lots of different spots in the genome. And so if you have high oil content, presumably you have a lot of dominant alleles where each dominant allele contributes a little bit to oil content. If you have low oil content, it means that basically you have very few dominant alleles. So if you're now selecting for high oil content, but you're starting out here at 5%, there's a lot of room for you to select for more dominant alleles. So these dominant alleles come up through mutation. The mutation leads to that plant having more oil content. Then you set that plant aside and you have it be the basis of the next generation of growing. So you have a lot of dominant alleles to get back into the population to raise oil content. So there's a lot more genetic variation to act upon than if you start with 5% and you're driving it down towards 0%, because all you're really trying to do is you're trying to get rid of the couple of dominant alleles that are there. Once you get rid of all of the dominant alleles, you're bottoming out. There's no place to go. You're at the bottom. All of the dominant alleles are gone, and that's what's happened with this continuous low oil population by the time you get out here to 80 or so generations. You've just depleted all the variation. All of the plants in this line at that point are this genotype for oil content. Before they got there, though, back here at about 50 generations, they reversed that and they said, this low oil content line, we're going to reverse selection. We're going to try and get more oil out of those. So we're going to start selecting for high oil content. And in the same way, at this point, they took the high oil content population and said, let's try driving it downward, select for low oil content. Once again, you see that this is increasing, but it's increasing at a very slow rate. It's got a really low slope. But this decreases, but that slope is much steeper. Once again, the more variation you have in the population, because that's what heritability is. Heritability is a measure of genetic variation. The more variation you have when you're trying to get your population to respond, it's due to selection multiplied times the amount of genetic variation that you have. This population still has a lot of genetic variation in it because you can continue selecting it up and it doesn't slow down much. And when you select it down, it goes down really quickly because heritability is high, which is another way of saying there's a lot of genetic variation in this population. At this point in time, the population isn't like this. It's probably more like this. About half of those alleles are there, which means that there's room to either go up or go down in terms of oil content. So they reversed it, and they found kind of what you would expect. In the lines that had their genetic variation depleted through heavy selection, it increased, but it increased slowly. In populations that still had a lot of variation, it decreased, but decreased quickly. And then they re-reversed this line here, said, okay, we've gone 10 generations of making it lower. What happens when we select for higher oil content again? And it reverses and it goes up at about the same rate as this is, but they're slightly different. And they're beginning to converge on one another here at the end. What that means is that they're probably approaching the upper range of oil content. So they're beginning to get to that point where you're moving closer and closer to this, that will then move closer and closer to this, that will then move closer and closer to this. And then at that point, these high selection lines will stop evolving the same way that the low selection lines will stop evolving. Does that make any more sense? Okay, good. That's the reason I wanted to ask you guys if you had questions. Somebody else had a question. I thought I saw another hand up earlier. The cool thing about this way of thinking about inheritance is you don't have to know the specifics of any given gene acting on this trait. All you need to know is, do parents resemble their offspring? And to what degree is that resemblance found? So if you've ever been at a party and friends of your family, or your family members for that matter, and they're like, oh, she's got her father's eyes, or oh, he has his mom's chin, or whatever. People are picking up on this resemblance among relatives, and that's what this is basically looking at. To what degree does now that just looks like I've drawn a gigantic penis on the screen. That's not so good. To what degree do the parents resemble their offspring? If that resemblance is strong, that means that that particular trait is highly heritable. You guys probably all resemble your siblings somewhat, and you also probably resemble your parents somewhat. It's not perfect resemblance because we go through sexual reproduction. We make gametes, but they only have half of the genetic components from your mom, only have half the genetics from your dad. Put together in you, that makes you who you are, and you're unique from both of them. In the same way, your sibling gets half of their genes from your mom and half of their genes from your dad, but it's a potentially totally different half than the two halves that you got, which is why you might resemble your siblings, but you're not identical to your siblings. This all comes about because we make our gametes via meiosis rather than some other method of cell reproduction. So we don't need to know what the genes are that are influencing height. All we need to know is, do tall parents produce tall offspring? Do short parents produce short offspring? Do average parents produce average offspring? Now, if we are going to think about this from the standpoint of like height, these are the gametes that come out of a cross between these two parents. This is an average height individual. This is an average height individual. When these two individuals go to reproduce, they can produce anything from average height individuals, all these threes scattered around in here, all these threes are average height. That's what theirs are. They have 3 dominant alleles each. But these individuals can also produce the tallest individual in the population, and they can also produce the shortest individual in the population. So you may not resemble your fellow siblings at all because you happen to get all of your parents' recessive alleles. Your dad gave you his little a, little b, little c, and your mom gave her her little a, little b, little c, and you ended up being homozygous recessive for these three genes, which made you the shortest of the short. But you could have had a sibling who got all of your dad's big alleles, all of your mom's big alleles, and you have a sibling that is this genotype, making them the tallest of the tall. And that could have happened from both of your average size parents. Now, the odds of that happening are really small because the odds of getting this is 1 in 64, and the odds of getting this is 1 in 64. So it could happen, it's just unlikely. But it could. But generally, we would guess that the bulk of the 64 is right here in the middle. It's not at the extremes. The bulk of the 64 is right here in the middle, not at the extremes. And so you're more likely to resemble your siblings because you all are all probably in this part of this distribution. Now, a farmer trying to improve his crop, whether it's improving milk production or oil content in corn or whatever, he's not interested in the middle. He's interested in making more milk, making more corn oil, making more whatever. So he selects against this end of the distribution in the hopes of driving his population, driving his crop to the end of the distribution that's going to make him the most money. This is what artificial breeding is about. This is how we get from here to here. From 5% oil content to close to 20% oil content. Is just every generation selecting those individuals that produce the highest oil content corn, allowing those to form the basis of the next generation, and then just doing this over and over and over again for 100 generations. Other questions about this screencast? Okay. The last thing that I wanted to talk to you about that I thought would probably be better to talk to you about in person than in a screencast is hybridization. So if you ever talk to a farmer, how many of you come from farming backgrounds? I think I've asked this already. None of you, right? None of you. If you ever talk to a farmer, you will find that most of them buy hybrid seed. And they buy hybrid seed in spite of the fact that it's very expensive because they think that it's going to help them get a higher crop yield. And the reason hybrid seed is expensive is because it's expensive to produce. Because what you have to do is you have to have large amounts of land to grow specific varieties of corn. So what you do is you select for drought resistance in a line of corn. So you'll have a cornfield or multiple cornfields that are just devoted to making corn as drought resistant as it can be. And you allow these corn to inbreed over and over and over again until they're finally kind of like the pea plants. They're homozygous for all of their traits. But one of the traits that they're homozygous for is drought resistance. Then you have another line that you select for pest resistance. And once again, you breed that generation after generation after generation until it's homozygous for all of its traits because you've inbred it so much. But one of those traits that it is homozygous for is pest resistance. And then you might have another line that is designed to be resistant against some type of fungus, and another line that is resistant against some type of herbicide, so that you can spray herbicide on your crop. It'll kill the weeds, but it won't kill your corn plant. And in each of these lines, you've bred them over and over and over again until the entire genome is homozygous. for whatever genes you have in your genome. But the important thing is that for this one that is pest resistance, you know that it's homozygous for that one. And all the plants in that field have the same genotype because you've allowed them to inbreed for generations and generations. Remember, in fruit flies, you can do this in about 25 generations. So it takes time to get these inbred lines. And not a pharmaceutical company, an agricultural company, when they get these inbred lines, they maintain these inbred lines. And then what they do is they breed one inbred line with another inbred line by detasseling one set, taking the tassels from the other set, and fertilizing the corn. And so you now have a hybrid crop. So if this is line B, you also have line A, and line A has the gene C, which is for drought resistance, but it is homozygous recessive now for pest resistance because they weren't breeding it for pest resistance, they were breeding it for drought resistance. Now when we cross these, we will get an individual that is heterozygous for drought resistance, but because drought resistance is a dominant trait, that's the trait that gets expressed, and it's now heterozygous for pest resistance, but once again, because pest resistance is the dominant trait, it's the one that gets expressed. And so now you have a strain of corn that is both drought resistant and pest resistant. You're doing the same thing for whatever two traits are over here until you get it into this line. Then you cross those with one another and you get a line of corn plants that now have all four traits in it. But note at this time that in this case, all four traits are going to be heterozygous. Now the nice thing about that is that they'll all display the traits that you want. But remember that if you're crossing an individual with a bunch of heterozygosity in it, each one of these genes that are of interest to you are heterozygous. If you then cross those, so let's say the farmer decides, I'm sick and tired of buying hybrid seed that is so expensive from Monsanto, I'm going to just let my plants freely breed. And save myself some money. What happens? Well, let's go back and just look at the 3-gene case. We would want dominant alleles. Let's say we were just looking at 3 traits, and it becomes more complicated with 4 traits. If we wanted 3 traits and we wanted to have at least one dominant allele in each of these, that's checked off here, 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 not here, 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 and here, of course, here and here, not there, here, 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 not there, here, here, nope, not there. Homozygous for the A's, da 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 da. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27. only 27/64 of that population would have those 3 desirable traits. Over half of the next crop that farmer got would not have all of the desirable traits that were in the hybrid corn that he originally bought because of simple genetics. So you ask yourself, why do farmers buy this really expensive hybrid seed to plant in their fields? It's because of simple Mendelian genetics. If they were to save the seed that is produced from their crop, hold what they call holding back seeds, which is technically against the law. When you buy hybrid seed from an agricultural company, you sign a form that basically says you're not going to hold back seed. But even if they did, it's not in their economic best interest because less than half of their crop resulting from holding back that seed would have the traits that they were buying that hybrid seed for in the first place. So farmers complain about the high price of hybrid seed, but they keep buying it because they don't have the means themselves to maintain these inbred lines, do the crosses. Imagine that you get 2 crops of corn a year. It will take you 2 years to do all those crosses. After having done all that inbred line work, getting these inbred lines formed to begin with. So Monsanto is thinking 2 years out as it's doing these crosses as to what types of trait combinations farmers are going to be interested in because that's how long it takes to do this. This becomes outside the realm of possibility for any normal farmer to do. And so it's just quicker and easier and quite frankly cheaper for them to just shell out the money for hybrid seed every year. Because saving back seed just doesn't work in the long run. Okay? How do we know this? Well, because we can do a Punnett square. Okay? And look at the outcomes of that. In this case, it isn't important how many dominant alleles. In this case, it's important which dominant alleles occur where, in which genes. But the trihybrid cross is useful for that. All right, questions about that? All right, hopefully at this point you know a lot more about genetics than you did when you first started. Have you guys been working through the genetics homework? Some of you have, the rest of you haven't. The rest of you need to do that if you haven't done that already. If you're having difficulties with it, come to class on Wednesday. If you have a question over any of those problems, we can work through them in class. But take a shot at doing those first before you come to class with questions. Feel free to come to my office and ask me questions. I have a big whiteboard in my office. I have a whiteboard in the hall outside my office. We can work through genetics problems. Genetics problems will be on the exam. The whole reason that homework is there is for you to practice these things, okay? All right, moving on. This is much more productive than me just re-delivering the lecture that's already on the screencast, in my opinion. All right. Oh, but then I robbed my glory. So all of this selection that we have acting on populations that's being exerted by farmers, all of this kind of selection like this is basically how we got this thing from wolves, which is where domestic dogs originally came from, and corn from teosinte. And I'll show you a picture of what teosinte looks like. So these are modern genetic varieties that came from natural plant progenitors. We're going to have an entire set of information coming up about where plants came from and what they looked like when they were wild. But modern corn comes from a plant called teosinte, and a modern pug came from a wolf. Both of these modern varieties of these animals that occurred out in nature are the result of lots of generations of humans selecting for particular traits. I do not know why you would select for the traits that are found in a pug, but there you have it. I will occasionally bring in memes because I find memes to be funny and irritating. A guide to identifying genetically modified corn. On the left-hand side here is teosinte. This is the original progenitor of what eventually evolved into corn. You can actually select on 7 different genes in corn, in teosinte, and convert it into corn. So major modifications at 7 genes take something that looks like this and turns it into this, as it turns out. What does GM stand for? You guys know? Genetically modified, yes. When people use this term, what are they usually referring to? This is an important distinction that I want you to make. The authors of your book chapters that you're going to read, they make this distinction, but then they're not particularly good at using the distinction in the chapters. But oftentimes, when people think of genetically modified, they think of I make a distinction between genetic engineering and genetic modification, and I will not be sloppy in how I use these two terms. And you should not be sloppy about how you use these two terms. This is genetic modification. Starting off with a plant that has a relatively few number of seeds, each of those seeds is bound up in its own little leafy capsule. Those seeds, when they're ripe, they will burst out of that capsule and disperse themselves. This is teosinte, which is the old form of what eventually came to be corn. But you got to this type of corn by farmers who started eating teosinte, saying, oh, here's a teosinte plant that has a few more kernels on it. I'm going to plant that one instead of this one that has fewer kernels. Oh, these kernels stay attached to the central stalk, the cob. As a result, they don't disperse, and as a result, I can just let it ripen on the cob rather than harvest it early. Or if I don't harvest it early enough, I have to pick up all those kernels off of the ground. Well, I'm going to plant the one that doesn't have what we call shattering seeds, these seeds that pop out and disperse themselves. I'm going to plant the one that doesn't do that instead. And over time, through incremental change, you get from this to this. This is genetically modified. It may or may not be genetically engineered. Genetic engineering is when we take a gene out of one organism, we cut it out of that organism's genome, and we splice it into another organism's genome. Now, that is a form of genetic modification, but it's a specific form of genetic modification that involves taking genes out of one species and putting them into another species. So it's a stupid meme. God, memes are stupid. You know what you should never look at the rest of your life? Memes. They're just pointless because in this image, there's so much complicated information that just gets thrown aside to make what somebody thinks is an amusing point. Because the meme doesn't discuss what's the difference between genetic modification and genetic engineering. And this is genetic modification, this is non-genetically modified, but that doesn't say anything about whether or not this thing has had a gene taken from a soil bacterium that kills insects inserted into its genome. And there's no way of knowing if this has been done to this plant by looking at it. That would be then a genetically engineered corn plant. Don't do memes. See memes on your Facebook page, or I don't know, what do you guys do nowadays? You guys don't do Facebook. How do you get your meme supply? Yeah, Instagram. Unsubscribe from Instagram, or at least those people who are commonly posting memes, because they just kind of dumb you down. And we want you to be intelligent, not stupid. Once again, genetic modification, selective breeding, choosing particular traits in wolves that people were interested in, eventually led to a pug. Piacente eventually got converted into corn. This happened over long periods of time. This is from Middens. How many of you are business majors? History majors. Oh, no history majors. History. Do you guys know what middens are? If I did not become a biologist, I might very well have become an archaeologist. Midden is just the archaeological term for an old trash heap. And the cool thing about middens is you can go digging around in middens, and basically every culture has had a midden. We have a midden here just south of the river. If you go on 291, you cross the river, you go up a hill, you go down into a valley, and there's a cement plant on one side of the road, and there is a gigantic landfill on the east side of the road as you're going down towards Independence. That is our midden. Every culture has had trash heaps, place where they put their refuse. In the Pacific Northwest, you can look in the trash heaps of the Pacific Northwest Indians, and you can see in the layers as you dig down through a midden, through a trash heap, you can see chronologically where salmon fisheries, commercial salmon fisheries started up. Because below that level, those trash heaps are full of salmon bones, and above that level, salmon bones are relatively rare because salmon stocks began to fall off precipitously after commercial fishing entered into the Pacific Northwest. And so a trash heap usually starts out in a pit, but eventually mounds up, and the top of the mound is the most recent trash, and the trash gets oldest at the bottom. And as you dig down through this trash heap, you get to older and older layers of people's trash, and that becomes informative. And so if you go to Mesoamerica, if you go to Central America, Mexico, and dig into old Mayan trash heaps, what you will find as you go down is you will find corn cobs. And the corn cobs get shorter and shorter as you go back in time, and they have fewer and fewer kernels as you go back in time. So over time, farmers pick the seeds that they're going to plant, but this type of artificial selection, although it's quicker than natural selection, it still takes thousands of years in order to get you to the end product here. Piacente is this multi-branched plant that you can still find growing wild in Mexico, but over time, once again, it became evolved into a single stalk. This had multiple flowers, and usually modern maize has one or two cobs on it. Go out to any of the cornfields growing around here. If you go out to a corn maze or something like that, look at the corn, you're going to find only one or two cobs on each plant. And they're located about 2/3 of the way up the stalk. So we've gone from a branch plant with many flowers to a single stalk plant with only two flowers, and from a plant with relatively few kernels to a plant with a lot of kernels, and those kernels are wrapped up in multiple layers of leaves so that those kernels can't get away. The plant can't disperse those kernels because the farmer doesn't want those kernels to be dispersed. He wants them to stay on the cob so he can harvest them without too much trouble. So this is the long progression of genetic modification. Farmers picking which seeds they're going to plant, getting the results of that, planting another select group of seeds, getting the results of that, and on and on and on for 10,000 years until you have corn, what we think of as corn. Is corn genetically modified? Yeah, it's gone through a long history of tens, 10,000 years of, or maybe longer now, we know, because of a recent discovery of some fossil footprints that are much older than we used to think, for people being in North America. But over 7 to 9,000 years, we made this we didn't, but Native Americans made this transformation from teosinte to corn, modified 7 genes, essentially selected for 7 genes that lead to this transformation. That's genetic modification. This is just from that same paper, looking at the types of genes that are found in teosinte versus maize, and you can see the kinds of genetic changes that have happened. These are the names of the genes that you can find in these panels down here, and you will see that the representation of different varieties of the gene do vary from teosinte to maize. You don't need to be very concerned about this. Just know that what led to corn becoming corn is genetic changes in a list of prominent genes. Genetic engineering is different, though. So let's talk about genetic engineering. Genetic engineering is when you take a gene out of one species and you put it into the genome of another species. And a lot of times we get these genes that we put into plants like corn and soybeans and stuff like this, we get them from bacteria. One of the reasons that we get them from bacteria is bacteria are super diverse and they do all kinds of cool things biochemically. And so the other reason that we use bacteria for this oftentimes is that we know that bacteria have certain characteristics that allow them to move genes from one individual to another. So one of the things that bacteria just naturally do is they'll cut out chunks of their own genome and transfer it to another bacterium. And so molecular biologists, cellular biologists, have basically taken the machinery that bacteria already possess because they're bacteria and adapted that capability to move genes around that the researchers want moved around from one individual to another. And so we have a bunch of different crops: alfalfa, canola, corn, cotton, papaya, potatoes, soybean, squash, sugar beet, and tomato. And here are a sampling of some of the genetic modifications that have been brought to these crops through genetic engineering. So a lot of these are herbicide resistance. The advantage of herbicide resistance is that you can spray herbicide on your crop. It will kill the weeds, but because you planted an herbicide-resistant crop, your crop doesn't die. So you don't have to have weeds competing with your crop plants because you can just kill it with the herbicide and the herbicide won't hurt your plants that you're interested in. There's a gene from a bacterium that kills insects that has been inserted into corn and cotton and potatoes. Genes that delay ripening so that a farmer can pick a tomato that is closer to ripening. It looks redder, it sells better in the supermarket, but the ripening process is slowed down so that it doesn't ripen so quickly that by the time it gets shipped to the supermarket and sits on the supermarket shelves for 3 or 4 days, it's overly ripe and starts to rot. So genes for delayed ripening, virus resistance, oleic acid production. So basically, soybean oil production has been improved in plants. So when we look at, this is old data from a textbook, but when we look at the proportion of crop that is genetically engineered versus not genetically engineered, for some crops it's much higher than other crops, and it just depends on the crop as to kind of how much crop is actually out there in terms of total crop that is genetically engineered. And then this breaks down kind of among the genetically engineered, what brand of genetic engineering is in there. Is it herbicide resistance, insect resistance, or a combination of the two? So we're almost out of time. The next slides go through history. So when did we first get this capacity for doing this? Um, we talked about RNA, messenger RNA vaccines, and how people are like, ooh, we're afraid of those because they're new. Well, they've actually been around since 1982. Just because you didn't know about it doesn't mean that it wasn't happening. It just means that you didn't know about it. Same thing with genetic engineering. Genetic engineering has been around for quite a while. Simply because you didn't know about it or think about it doesn't mean it wasn't here. It just means that you didn't know about it. So we'll go through kind of the history. Then we're going to go through actually what's happening at the level of the genes and how it works, how scientists actually do this. And then we're going to talk about an actual case that has happened over the last couple of years. There's this new technique called CRISPR that you may have any of you heard of CRISPR? Okay, a couple of you have heard of CRISPR. CRISPR is a new method by which scientists can manipulate genes, and they can do it in real time in an actual person. And there is a woman who has sickle cell anemia who has been CRISPR'd by scientists, and there are a couple of National Public Radio programs about CRISPR being used in humans. There are links to those radio interviews on the Moodle page. I want you to look at a video that describes how CRISPR works, and then right below there are the two links to the NPR stories about this woman who has basically very possibly been cured of sickle cell anemia, which is a single nucleotide polymorphism that occurs in hemoglobin that leads to this genetic condition where your blood cells don't function the way they should. And they basically killed all of her, they harvested her stem cells that are responsible for creating blood, for creating red blood cells. They CRISPR'd them. Then they killed all of her blood creating stem cells in her. Then they put these stem cells back into her, and now her stem cells make normal blood where she was making sickle cell anemia blood. And it's pretty freaky weird and cool. So between now and Wednesday, go and look at those materials about CRISPR, because we will definitely be talking about CRISPR on Wednesday. Questions? All right, we will see you on Wednesday. Oh, I went off and left the bag with your food journals in it at my house this morning when I left. When I get them back up, I'll drop you guys an email and you can come by and get those. I took them home to read them, and then I just forgot to get them in the car this morning.
Will you still need like today?
No, don't worry about today. You'll get them back, and when I read them again, I'll remember that I was suffering a brain fart today.
The chapter's at. Where the chapter's at on here?
So if you click on the folder, that's the document. And then if you go down a little further, this is the CRISPR stuff down here. And those are all just links. When you click on them, it should open the link in a browser. Yep.
The chapters are in.
Are in the National Academy of Science folders. So when you see a folder here, that means that there's a folder that has stuff in it. In this case, the folder just has one file in it. Download that file. That's the whole book. Other things, it shows that there's a little like chain on the side. That refers to a link that's linking you to something outside the course. And that's what the CRISPR videos are about. Yes, sir.
Should we be reading anything out of all the words of or just focus on
No, we're still in chapter 1. So all of this stuff that we're doing right now is basically getting at information that is in chapter 1. So if you've forgotten that last end of chapter 1 that is about corn, sex, and genetics, go back in
chapter 1, like chapter 7, when he's got the McDonald's.
Yeah, go back and reread that chapter 1 again if you've lost track of why we're still here. Because there's so much science packed into chapter 1, it feels like we spend forever in chapter 1 and then we blow through the rest of the book. And some sections of the book we don't even pay attention to. But there's just too much science in chapter 1. and he does such a poor job of delving into it in any kind of depth that it's hard to understand what he's talking about if we don't do all the background. So no, we're still in chapter 1.
Okay, that's good. I was like,
Yeah.
I was worried that like people were like, yeah, like chapter 10 or something.
Nope, nope. When we move on to the next chapter, I'll definitely let you know.
Okay, so